हॅलो 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 आवाज माईक नाही असू
Hello, hello.
हॅलो A very warm welcome once again. A very good afternoon to everyone. Myself Minal Rao, Assistant Professor at Adarsh Institute of Pharmacy. I hope you all had a refreshing lunch break and feeling refreshed for the upcoming session. This event is not just the meeting of mind, it is a collaborative platform to catalyze change in healthcare through innovative research and sustainable pharmaceutical practices. To grace this conference, we have esteemed dignitaries from the pharma fraternity. Dr. Vinod Thakre sir, Principal, Nagpur College of Pharmacy. Dr. Sachin Hiradeve sir, Professor, Nagpur College of Pharmacy. Dr. Vaibhav Uplenchiwar sir, Professor, Nagpur College of Pharmacy. And Dr. Jignesh Doshi sir. Now, I would like to request Mrs. Anshu Dudam to kindly come forward and felicitate our honorable guest, Dr. Vinod Thakre sir. I would like to request Mrs. Shabnam Seher ma'am to kindly come forward and felicitate our honorable guest, Dr. Vinod Thakre sir. Please sir. त्यांना वरतीच बसायला पाहिजे. Now I would like to request Dr. Sachin Hiradeve sir to please come forward. And I like to request Mrs. Shabnam Seher ma'am to felicitate our honorable guest, Dr. Sachin Hiradeve sir. Now I would like to request Parag Charde sir. Is here? Parag Charde sir to kindly come forward and felicitate Dr. Vaibhav Uplenchiwar sir. It is my great honor and privilege to introduce our esteemed speaker for this session, Dr. Jignesh Doshi sir, Director of the Center for Interdisciplinary Research and Development at GH Raisoni University. Dr. Doshi sir is a distinguished scientist with extensive experience in both academic and pharmaceutical industry. His expertise lies in synthetic organic chemistry and chemical biology, with remarkable contribution in the design and synthesis of small molecules for understanding complex biological systems. His research in green synthesis and biogenic nanoparticles for antimicrobial and anti-cancer application is truly inspiring. He has also contributed significantly to drug discovery during his tenure at Advinus Therapeutics, and his research work has led to prestigious international publications and patents. His academic journey, including his research at University of Minnesota, reflects his deep commitment to scientific excellence. Now, I would like to request Ms. Kirti Chadha ma'am to kindly come forward and felicitate our honorable speaker, Dr. Jignesh Doshi sir. Please, sir. Now, I like to invite Dr. Jignesh Doshi sir to deliver his valuable insight with us. Please, sir.
I have given my presentation.
Yes, sir.
स्लाइड चेंजर भी इसी में है.
हाँ.
ठीक है. अदरवाइज
उसे लाया देगा आपने?
हाँ मैं. ऑल राइट. फाइन.
सोबत राहिल की वायरलेस
नहीं वायरलेस कनेक्ट कर दीजिए.
शुरू वायरलेस
ये पॉइंटर भी है, ये पॉइंटर
बटन बनना है.
पॉइंटर है चार.
अच्छा उस पर रिफ्लेक्ट नहीं होगा. ठीक है. नो इश्यूज.
बाकी है चार. बाकी और है.
ओके फाइन.
ठीक है.
वायरलेस
नहीं दिस विल बी ओके. So, thank you ma'am for the introduction and very good afternoon to all of you.
हो जाएगा.
So, very good afternoon to all of you. Session after lunch is actually very challenging, right? Yes. I truly understand and I also empathize with you. Because like you, I was also a student of B.Pharm and I remember my days when I attended conference. After lunch session is challenging, but now today my goal here will be to try to share with you some of the latest developments of where AI is going with regards to pharmacovigilance. And I'll try to make my talk interesting so that it is not a challenge for you to stay awake, right? Fine. So, as you know, the title is AI in Pharmacovigilance, the transition from reactive safety to predictive intelligence. Fine. So, why are we studying? Why is this entire conference about pharmacovigilance? Okay, I will try to keep my talk in a way that I will ask questions. आप लोग आर यू ओके आंसर देने के लिए? अच्छा, वन मोर थिंग, इंग्लिश हिंदी मिक्स और प्योरली इंग्लिश? मिक्स. ठीक है. फाइन. सो या तो ठीक है. फाइन. सो एनीबडी वांट्स टू आंसर दिस क्वेश्चन, व्हाई आर वी स्टडिंग, व्हाई आर वी हियर टुडे फॉर द टू डे कॉन्फ्रेंस ऑन फार्माकोविजिलेंस? व्हाट इज द इंपॉर्टेंस ऑफ दिस सब्जेक्ट? अच्छा, नॉट एबल टू हियर एट द बैक. Okay, I think this is better. So, anybody wants to share their thoughts? हम लोग क्यों pharmacovigilance पढ़ रहे हैं? Why are we studying or why are we even thinking about this? Answer is there right here, right? So, every year, thousands of ADRs. So, what is ADR? Yes, great. Sir, objective achieved. So at the end of the conference, students are at least knowing ADR. So adverse drug reactions, right? So every year thousands of ADRs are going undetected. And that is a major reason why we should focus on pharmacovigilance. So what exactly is it, right? So as I've said here, a drug story does not end when a prescription is filled. जब आप काउंटर से मेडिसिन लेते या any prescription is delivered, that is just the beginning of the story, right? Because then what happens after taking the medicine, if there is an adverse drug reaction, then that leads to the entire events, right? So what is pharmacovigilance? As all of you are aware by this time, so it's a branch of science which deals with detection. Okay, this order is important, right? First what you do is detect, assess, understand and then prevent, right? पहले ADR होता है तो आपको detect करना पड़ेगा ना. Once you need to understand कि ये ADR है, the patient is having an adverse drug reaction, has to be detected. Then you need to assess it. Why it is happening? What are the other medicines? Is this a drug-drug interaction or is it a genetic? Genetic इसका cause है या इसका cause क्या है? वो assess होगा, right? फिर उसको और understand करने के बाद, then the next line is to prevent it, right? If this is happening, next time this particular class of molecules or this particular therapeutic drug class में, this should not happen or we need to prevent. If we are knowing that some ADR is happening, so then we can prevent it in future. So that is the entire circle of pharmacovigilance. Okay, the pointer will not show on this anyway. So why are these important? ADR क्यों important है? Why do we need? ठीक है, हो रहा है तो if it is 1 in a million या 1 in a billion एक जन को हो रहा है तो then we would not worry. But the statistics are shocking. So top 10 leading cause of death is due to ADR. So this is a very shocking statistic. And this is also responsible for 2.7 to 15.7% of all hospital admissions. Means a patient ending up in a hospital, so 2.7 to almost 3 to 16% of the times it is due to some adverse drug reaction. And finally, in case of other patients who are just admitted to hospital for some other reason, in those patients also, in 17% means 17 out of 100 patients will experience some form of adverse drug reaction. यानी ये जो reasons हैं, इसके कारण, these are the reasons why studying adverse drug reactions and studying pharmacovigilance is important, right? So challenges क्या है? In terms of pharmacovigilance, pharmacovigilance में challenges क्या हो सकते हैं? Like I said, जो उसका definition है, उसी में से के challenges भी है. पहला challenge, detection. Most of the times, a patient or a person who's taking the medicine is not able to detect. Sometimes they don't see that as an adverse drug reaction. वो सोचते हैं कि this is a normal reaction or a normal phenomena. I have taken this new medicine and maybe, I am sweating or I have some itching or a rash. So it is considered, patient may consider it as a normal reaction and it will go undetected. So the first major challenge is detection, delay in detection. Maybe they are taking the dose for a long duration, say anti-diabetic or some drug like that, anti-hypertensive. Taking it after 3-4 days or 1 week or 2 weeks, they start developing some irritation rash. So delay in detection. पहले दिन हुआ था but ignored. So delay in detection. Second is drug-drug interaction. Again, already on some medication, new medication has been started. वो दोनों drugs का कुछ interaction हो गया है and that is causing an adverse drug reaction. So that also is. Then the third challenge is from terms of people who are studying, not from the patient side, but from the people who are studying the subject, like from the clinician and from the researcher point of view. And that is coming from data volume. So a lot of data is there. Hundreds and thousands of patients are taking the medicines. And in hospitals and all such settings, a lot of data is being generated from diverse population. So heterogeneous data is there. So that also is a challenge. दूसरा, fourth one is a manual review. So अभी तक currently what we are doing, what is the tradition? Patient files are reviewed manually, entry is done manually in hospitals, and all the records and the review is done manually. So that is also a big challenge considering the huge number of patients. Then data integration and regulatory compliance. So data integration, again, it is related to the data itself. How to integrate the data with the patient's background, health background. अच्छा, do you know the health records, electronic health records, EHR, सुना है क्या term? नहीं? Some people are saying yes, some no. So electronic health record is kind of, as it's very simple, जो name जैसे उसका है term, आपका entire आपका जो health record including your age, weight, all the things and your current medications, everything in one digital form, right? So that has to be, that integration of all that data is also a big challenge. And then next is regulatory compliance. So basically all the regulatory agencies who are monitoring, acha, phase 4 everybody knows, right? Phase 1, phase 2, phase 3 or phase 4, right? Post-market surveillance, right? So that is the part where regulatory bodies are involved. So these are the challenges. And what about the data sources? The data, ये data कहाँ से आता है? From various sources, right? From clinical trial database, from electronic health records, which I just explained. Then papers, scientific papers are published wherein people are discussing about, see, once the clinical trial is done, uske results bahar aate hain, right? The drugs, the possible side effects and all that. If at all a drug is not, is failing in the phase 1 or if failing in phase 2. So those studies are published. It's mandatory to publish those studies so that people are aware. So that is the scientific literature. Then social media platforms also nowadays very, very active. If a person is taking, say, it's a approved drug. I am talking about an approved drug, right? Not something under clinical trial. So approved drug hai, if somebody is taking it, or let's say a topical application, oh, they are using that cream, then social media pe they are sharing with their friends, no, don't do this, I got this rash, you know. And all sorts of such people are nowadays putting their comments and their reviews. So social media se bhi, you can get the data about the various drugs and the adverse effect of the drugs. Acha hai to wo bhi aayega. And if there is something wrong, then also people will nowadays put it on the social media. Then claims database is related to the patents, wherein patents mein people are claiming that this particular formulation is, you make a claim that it is designed to cause this or it is designed to improve this particular disease. So that is from the claims database. Then genomic database, wherein different people with different genetic composition. In this hall, we are all having different genetic composition, right? Some people may be allergic to some particular class of drug. Many people are allergic to sulfonamides या sulfa class के जो medicines होते हैं. आजकल ज़्यादा use नहीं होते हैं. But earlier when I was your age, they were still very used. So then that is a genetic kind of a genetic basis for an adverse drug reaction. So genomic database also serves as a source, right? And then the last one, if you go up, it is spontaneous reporting system, which currently is very weak. When you go, if some of you practice as pharmacists or hospital pharmacists or general pharmacists at a shop, retail pharmacist, then how many times have you seen, or if your seniors are working, how many times have they seen that the patient himself or herself goes and reports? This awareness is not there. That, okay, I have taken this medicine, and sometimes it may be very simple thing that the color used in the tablet may be a cause of a reaction, allergic reaction. So there are very minor things which can lead to adverse drug reaction. And now the last thing is that various AI-based frameworks which are now being used to solve these challenges. So I'll just highlight couple here, and then as we go further, we'll discuss some of them. So first is a natural language processing. So natural language processing मतलब क्या है? हम लोगों का humans का natural language क्या है? Whatever English, Hindi, Marathi, whatever vernacular languages, right? But does a computer speak that language? Does it? No, right? So, and the notes, patient के जो notes है, doctor के जो patient के notes है, या जो भी है, the literature, scientific literature, it is published in a natural human language. That has to be converted into a language which can be understood by the computer and which the computer can then analyze, right? So that particular task is a task of AI, which is called as natural language processing. Then multimodal data fusion. So data is there scattered from different sources, how to integrate and fuse it and get them.
And then deep learning. So deep learning is also a type of AI-based technique wherein, like our brain, so in our brain we have neurons, everybody is aware, right? So likewise, in the computer also, there is something called as a neural network. Neurons to nahi hote hain, but there is a network of, there's a network by which the information is flowing, like in our brain. So that is called as a deep learning, and that is, the neural network is a part of the deep learning. So, and then predictive analytics. So predictive analysis is based on whatever information is fed, it will then predict that what will be the outcome. So if you are feeding the information of 1,000 patients, when they took this medicine, 200 or 100 developed this symptom, or 20 developed the other reaction. So based on that, and with different age group or different underlying diseases, से किसी को कुछ और एक साथ 2-3 डिजीज हैं, सो पेशेंट विद से सीवीएस कार्डियोवैस्कुलर डिजीज, 1000 पेशेंट में से 25 को सीवीएस कार्डियोवैस्कुलर डिजीज था, एंड देन दे आर शोइंग सम अदर रिएक्शन. सो दैट based on that information, the computer can predict that in future, if a patient is having this cardiovascular disease, then he or she may be at risk for adverse drug reaction. So that is called as predictive analysis. So now, if you compare traditional pharmacovigilance versus AI-driven pharmacovigilance, right? So there are some parameters which I have shown here, and then what is the difference? So first is the method. So traditionally what people do, those who study pharmacovigilance, they manually prepare charts, they review and they prepare their assessment. Needs expertise, takes a lot of time. If you do it through AI, what happens? It is automated, right? Manual component is removed. It is a continuous process, and using natural language processing, which I just explained some time back, what you can do is extract all the information from the patient records, and immediately or within a very short period of time, you can generate the entire database. Second is timing. Timing of wherein the pharmacovigilance role plays. So traditionally it is retrospective. Retrospective matlab event hone ke baad, when people are getting some kind of an adverse reaction, somebody is reporting a drop in a BP or a rash or a weight loss, XYZ as a side effect, only then people are looking into it that, okay, what could be the possible cause, what is the mechanism, what was the drug which is. So review is occurring after the harm occurs, right? Predictive matlab, you can do the analysis before the risk, before the event actually happens. So risk analysis जो है वो आपका event होने के पहले ही, based on all your machine learning, you can predict that, okay, if this is the condition, then the person may get this. This is, I have explained this in a slide further, so that time it will be clear. Now data scope. So data scope क्या है? इसका sample based, right? So जितना sample रहेगा उतना आप उसके ऊपर ही कर पाओगे manual में. When you are doing, if you don't get the data, जैसे I was telling, if people don't report, on day one they are seeing some abnormality in their body due to the new medicine, but they are not reporting, so that data is lost, correct? But इसमें कैसा है कि even if people are not reporting, their electronic health records are available through the hospitals, they are shared, and through the EHR, data can be extracted. EHR में क्या होगा कि अगर patient का BP drop हो रहा है after taking the new medicine, it is recorded in the electronic health record, and the patient is not even aware, but the data is extracted by AI and it is getting there. Then scalability, simple है. Human का जहाँ आता है तो we have a limitation, right? Most of the human beings, they are working in offices for 8 hours. उसमें से भी seriously कितने hours? Whatever, up to their personal capacity. So there is a limit. Then number of manpower, you can only have a limited number of people, experts who can work on it. But in case of an AI, you can scale it to infinite, and the AI is like it's a software based thing, right? Computer based. So it can work 24/7. It doesn't need any break or any downtime, right? Last is the causality assessment. So causality means what? Cause the factor. What is causing it? So uske liye bhi there are algorithms called as Bradford algorithm or Naranjo algorithm. This can be done by experts who are trained in this. Ki what is causing this? Particular ADR if it is seen, what is causing? Uska assessment kaise karna? That is a expert thing. But now if you go for the AI method, then this can be done by again software which will work on probabilistic reasoning and pattern recognition. Pattern recognition matlab ki it will, based on all the data which it has in its own database, it will try to see if there is a pattern in the जो ADR हो रहा है and the observed the input data. If there is some pattern or correlation, it will recognize or identify that, and it will increase the speed of detection, right? So next is, so what is the typical workflow when we consider that AI in pharmacovigilance? The typical workflow kaisa hota hai? It is, I am just showing on this slide here. First one is the extreme left, you can see the yellow thing. So data is first collected from various sources, as I discussed earlier. सभी जगह से data लेते हैं. Data किस form में है? Human language, right? So it's in the form of natural language. Then it is subjected to something called as NLP, natural language processing. उससे क्या होता है? That is converted into codes. Codes are understood by the computer, right? Computer cannot understand the human language. So codes, एक बार codes में convert हो जाता है, then comes the machine learning. So the computer or the machine which is called here in this case, it tries to understand the information presented in the form of codes, and then it will try to recognize if there is a pattern, if there is some similarity. So that is the ML part. And then signal. So what is the signal? उसमें से whatever insight वो निकाल रहा है, उसका understanding का जो हो रहा है, that is the signal which it is coming, output जो है उसका. Okay, if this is there, then this is the output. That is there. And then finally उसका decision. So based on all the output, what decision needs to be taken, whether this medicine should be withdrawn from the market, or whether label should be written that such patients or people having hypertension or some other disease should avoid this medicine. ऐसे वो ultimate decision is, ultimately that is the decision. Now if so have you understood this, or it is looking too fancy? So if it is looking too fancy, I will present the same in trying to present it in a simplified version. ठीक है? 4-5 steps था तो उसको अभी एक step और कम कर दिया. Again, very first step is what? Clinical notes, reports. जो भी reports है, notes है, side effects के जो reports है, ये सब manual form से you are converting it, you are collecting it and converting it into what? Using NLP, because here what is happening, there is too much information. So the single person or human beings, when they are dealing with thousands and 10,000s of research papers or clinical notes, we have our limitation, है ना? We get tired and we get confused. So what is the rescue there? NLP. Natural language processing क्या करता है? Data extract करता है, normalize करता है. Normalize मतलब you need to average it, or you need to have a, what is the data is from different sources. बहुत जगह से है. कहीं कुछ पेशेंट का वेरिएशन है, कहीं कुछ जेनेटिक वेरिएशन है, लोकेशन वेरिएशन है, डिजीज कंडीशन वेरिएशन है. तो ये सब चीज़ों को नॉर्मलाइज करने के बाद उसको प्रॉपरली फॉर्म में कन्वर्ट करता है. सो दिस पार्ट वेयर ह्यूमन बीइंग इज स्लो, द कंप्यूटर डज इट मच फास्टर. देन अगेन आफ्टर द डेटा इज रेडी, व्हाट यू डू इज you mine the data and you detect some pattern. वो data को you मतलब the system does it. If human being tries to do it, it becomes very difficult and time consuming. But if the computer does it, it does it much faster. And it finds in thousands and 10,000 number of patients, it finds the data, it recognizes the pattern, and then this is the where the pattern recognition and the correlation is done, right? इसके बाद पैटर्न एक बार रिकॉग्नाइज हो गया तो फिर इसका इफेक्ट, फाइनल इफेक्ट क्या है? बेनिफिट टू द पेशेंट इन टर्म्स ऑफ अ, so here it's written signal, but signal is what? In terms of a kind of a warning or a kind of a message to the patient that okay, this is the pattern that has been observed in 1,000 patients in this particular Asian Indian ethnicity, in which females में इतना है, males में ये drug का ये reaction है. So likewise that entire the signal is generated as an output, and this safety signal is used by scientists, patients, clinicians, everybody, right? So now I'll just discuss couple case studies where, as I was telling you, that it will be better, I mean you will be better able to understand this. So this is a research paper in which the developers, again these people are, देखिए ये लोग कौन है? Computer software engineers, right? We are not software engineers. I am not. Some of you might be knowing or some bit of software, but we don't know as much as the software engineers do. So what they did is they designed an AI-based system for medical literature monitoring. जो भी medical papers about the clinical trials and research articles जो निकल रहे थे, उन्होंने 10,486 research articles ये AI model ने में feed किया, okay? Of which what happened is 8,400 were used to train the model. So training we all know, right? Abhi, I think I should ask questions so that you people can. Training word का meaning पता है किसी को? उसमें IT और software वगैरह किसी चीज़ का रोल नहीं है. What is training? Anybody? ट्रेनिंग क्या होता है? सीखना, ठीक है, गुड. और कुछ? From this side, anybody wants to share? Okay, when you were kids, आप लोग जब छोटे थे साइकिल, अच्छा, how many of you know how to ride a bicycle? Excuse me. हाँ? साइकिल चलाना किसको आता है? सभी को? तो कैसे सिखाया गया था? एक जन बताओ. हाँ? So I think to answer this also you are discussing amongst yourself, हाँ? यानी बाजू वाले को पूछ रहे हो क्या? That you only can answer how you have been taught how to. You were trained some way using those. There is a bicycle with 2 small wheels on the side. ऐसा किसी ने सीखा होगा, या किसी के केस में mother or father might have, साथ में पकड़ के ऐसे पीछे. So you were trained, right? Some kind of a training. Even when you are doing B.Pharm, doing pharmacy, you are being trained here, right? Training मिल रहा है कि नहीं? Formulation कैसे बनाना या chemistry का reaction कैसे करना. So this is, you need to train things, humans also. So likewise, the software also needs to be, the AI system also needs to be trained. तो 8,400 articles का जो data था, that was fed into the computer, and whatever information was there regarding the adverse effect of the drugs or the so that information was used by the AI system. Based on that, it understood, okay, ये data है, ये ऐसा graph है, ये number है, that means it is a ADR, या ये BP high हो रहा है. Whatever information was there, that was fed in the computer and it was used to train the AI system. Then what they did is, उसके बाद they used 2086 articles, जिसमें there were some false articles, मतलब जिसमें nothing, no information related to any adverse drug reaction. There there is no information related to medicine also. ऐसे false positive बोल सकते हैं, ऐसे false entries भी थे 2000 में. There were some which were not discussing anything related to pharma or medicine, or such articles are also used, and they were used to test the model if the model is effective in picking out. So what they found, the study found that AI could successfully filter 55% of irrelevant articles. यानी 100 अगर irrelevant articles है तो उसमें से 55 irrelevant article AI का जो उनका डेवलप किया हुआ मॉडल था वो फिल्टर कर पा रहा था. It was able to identify, and it also identified 99% of the important articles जिसमें ड्रग का, सेफ्टी का, ये इट्स ऑल डन बाय कीवर्ड बेस, राइट. रिसर्च पेपर में अगर सेफ्टी, एडवर्स रिएक्शन, ये सब कीवर्ड्स वो स्कैन किया होगा सॉफ्टवेयर ने, सिस्टम ने, एंड इफ इट फाउंड सच थिंग्स इट जस्ट separated them. So this is how this system was, so this paper was published, and this system worked very well. 99% of the important articles related to safety were picked up. And whenever there was a doubt, अगर सॉफ्टवेयर को डाउट था तो it would pick it up, and later human beings can. So imagine here 10,000 research articles को human being को अगर पढ़ना है तो how much time a human being will take? Any guess? Guess कर सकते हैं आप लोग? 10,000 रिसर्च पेपर पढ़ने को कितना टाइम लगता है? हाँ, अगर एक दिन में 10 भी पढ़े तो राइट, ह्यूज. एक दिन में 10 पढ़ा तो 1000 days. And how many days in a year? And how many days we feel like working or reading? 1. Huh? नहीं ना? Fine. So this is what I am trying to say here in this case study. The developers, they developed an AI-based system, and they showed how it can be used to extract the information, and how it can, and how they were successfully able to develop this AI system which was able to filter out junk जो है 55% उसको ये कर दिया उन्होंने, and 99% of the time of the important articles were kept. Okay, second case study is about the NLP which I was saying. लेफ्ट साइड में क्या है, एक डॉक्टर ने नोट लिखा है पेशेंट को देख के. After checking the patient, inspecting or examining the patient, the doctor has made a note wherein the doctor has written that patient presented with severe urticaria. हाँ, urticaria क्या है? करेक्ट. अब बाकी को हाँ, रैशेस. सो आई वॉज गोइंग टू से हाइव सबको नहीं समझेगा तो रैशेस, राइट, सिंपल लैंग्वेज में रैशेस. सो पेशेंट शोड रैशेस. कब हुआ रैशेस? 12 hours आफ्टर टेकिंग विच मेडिसिन? सिप्रोफ्लॉक्सासिन, राइट. सो डॉक्टर हैज नोटेड दिस. नाउ एनएलपी ने क्या किया, ये नोट को कन्वर्टेड इनटू अ कंप्यूटर अंडरस्टैंडेबल आउटपुट. उसने किसको लिंक किया? हाइव्स, अर्टिकेरिया को लिंक किया एडवर्स रिएक्शन से. ये एक टाइप का एडवर्स रिएक्शन है. उसका ऑनसेट टाइम व्हेन इट स्टार्ट्स 12 hours. एंड थर्ड, सस्पेक्टेड ड्रग, कौन से ड्रग के कारण ये हो रहा है. सो ऐसे करके डॉक्टर के नोट को द सॉफ्टवेयर हैज कन्वर्टेड इनटू अ स्ट्रक्चर्ड फॉर्म. नाउ लाइकवाइज इमेजिन हॉस्पिटल में कितने सारे डॉक्टर्स हैं, उनके कितने नोट्स हैं. सो व्हेन ऑल द डेटा इज फेड, द एआई सिस्टम विल इमीडिएटली मेक अ वेरी नाइस डेटाबेस. उसके पास पूरा ये स्ट्रक्चर्ड इंफॉर्मेशन हो जाएगा, जिसके कारण नेक्स्ट टाइम इफ समबडी एक्सपीरियंसेस अ हाइव या अर्टिकेरिया, तो इमीडिएटली व्हाट दे विल चेक, कितने आवर्स के बाद हुआ और उसका मेडिकेशन में क्या सिप्रोफ्लॉक्सासिन था क्या, राइट. सो दिस इज द एडवांटेज ऑफ दिस सिस्टम. Again, another case is where I was talking about prediction, how AI can be used to predict the risk. So on the left panel, if you see patient profile, यानी उसका genetics, उसका electronic health records, lifestyle, whether the patient has a active lifestyle, whether he or she is very active या sedentary, बैठे रहते हैं, है ना. So that lifestyle, what kind of what are the other activities they are doing? Are they doing some exercise or it's just mostly sedentary? Then demographic. Demographic में मतलब their age, their gender male है female है, and the other उनका जो location है उनका, these things are whether their ethnicity, race, Asian है, African है, because you all have studied as per our genetic composition we have different ability to metabolize drugs, right? है कि नहीं? ये एग्जाम्पल पढ़ाया होगा ना कभी बायोफार्मास्यूटिक्स में. सो सम पीपल आर फास्ट एसिटिलाइटर्स और ऐसे अलग अलग हम लोगों के जेनेटिकली वी आर डिफरेंट पीपल आर रिस्पॉन्डिंग टू मेडिसिन इन डिफरेंट वे. सो ऑल दिस इंफॉर्मेशन इज फेड इन टू द कंप्यूटर सिस्टम, द AI मॉडल. what they do is they apply something called as neural networks and algorithms predict. उसके basis पे वो process करके they are able to make some prediction. What is the prediction? So here if you see on the right side AI analysis and prediction, उसमें वो they are showing that this particular patient is at high risk of ADR. Suppose if they find based on the information जो भी patient का electronic health record है कि वो already the patient is diabetic, वो ऑलरेडी बीपी के मेडिसिन भी ले रहा है और आप उसको तीसरा मेडिसिन कुछ और दे रहे हो. सो बेस्ड ऑन दैट द पेशेंट इट इज एट हाई रिस्क, स्पेशली इफ द पेशेंट इज सफरिंग फ्रॉम हाई बीपी, उसके किडनीज आर ऑलरेडी अंडर स्ट्रेस और आप मेडिसिन ऐसा दे रहे हो विच इज रीनली एक्सक्रीटेड. सो द पेशेंट इज एट हाई रिस्क, उसको रीनल फेलियर होने का बहुत हाई रिस्क है, ऐसा प्रेडिक्शन वो दे सकता है. देन the software can also tell you another thing. In the lower panel if you see preventive approach, so based on that computer is calculating कि ये data के हिसाब से maybe after 4 months of therapy ये के patient का kidney failure हो सकता है. So that is in the red dot, right? This pointer may not work because on the black screen. No, it's not. So that red dot if you can see, so if the prediction is at around 4 months, then they can also say that at 2 months you need to again check the patient, the kidney function, and see if there is some problem, adjust the dose, reduce the dose or change the medicine altogether. So this is the prediction of it, of the predictive risk model जो है वो ये है कि आपके जो data है उसके basis पे they are able to, the software itself is able to predict when adverse drug reaction may happen, if it will happen and when it will happen. Right? So what it is doing, it's helping to detect the risk early, fast. Then according to the input, the patient's data, it is able to predict the personalized safety for the patient, whether this new medicine is safe for the patient or it may cause some adverse reaction. And if at all it is at the risk of causing adverse reaction, then they can also suggest a preventive action कि आपको अगर ज़रूरी है ये medicine देना, for example you have to give, then if it is predicted after 5 months the patient will have adverse effect, so you give for 1 month or 2 months and monitor, and if need change the drug or stop the medicine. So having said that, so what are the challenges in AI based system? AI based system किस चीज पे डिपेंडेंट है? डेटा. जो भी डेटा इनपुट करते हैं अगर वो डेटा इनकरेक्ट है या गलत है तो आपका आउटपुट गलत होगा, बिकॉज आर्टिफिशियल इंटेलिजेंस इज नॉट एज गुड एज ह्यूमंस, राइट? इट विल जस्ट फॉर इट इंफॉर्मेशन इज इंफॉर्मेशन, वो लेके उसको प्रोसेस कर लेगा. Software will process it and give you output. But if you feed wrong stuff, output will be wasted or wrong. Next is the black box effect. So black box effect क्या है? Many times in many of the softwares we don't know what's happening. What you are doing is you are feeding the data, computer is processing it and giving you the result. But how, what, that we are not knowing. So for example in this case patient data is fed, output क्या दिखा रहा है वहाँ red box में? Infection risk of 85%. ये सॉफ्टवेयर ने बता दिया based on the data processing and all that. But how, what, that information is missing. So this is also a challenge in AI which is currently being worked on, and to quite some extent this has been worked on. And the next one is regulatory acceptance. Again regulatory bodies जो है हमारे FDA, US FDA है, इंडियन गवर्नमेंट का FDA है, CDSCO है, तो these are human based, they are not computer based or right AI based नहीं है ये. So they have some hesitance. They are not readily accepting AI based analysis. So that is kind of one of a challenge. Now how this challenge can be overcome? There is last couple years, 2 to 3 years back, there is this concept called as explainable AI or XAI. So what they do is, इसमें एक SHAP करके framework है. Again, framework is a kind of a algorithm or a software which what it does is quantifies each feature. यानी आपने जो भी डेटा दिया है, डेटा में से एक एक फीचर को फाइनल आउटपुट के ऊपर किसका कितना कंट्रीब्यूशन है, that it is able to detect. Like for example, जो the case which I discussed on the previous slide, डेटा देने के बाद 85% इन्फेक्शन आउटपुट आया, but if you consider on the right side panel, using this algorithm SHAP, it is able to say कि patient has prior antibiotic use जिसके कारण 40% risk बढ़ा, the patient has ICU admission जिसके कारण 25% risk बढ़ा, and then also patient's age is more than 65, which has led to a 20% increase. So total 40, 20, 25, total 85% का breakdown is now, so now no longer it is a black box, right? From a black box it has become a glass box wherein you can understand and you can each component's contribution to the output is explained. So now, so we are talking about the human, and I also explained about the AI-based pharmacovigilance and human-based pharmacovigilance. So if we compare these, so human-based क्या है, intuition के ऊपर based है, जैसे हमें अंदर से feel होता है कि नहीं ये this is a case of adverse reaction, and that comes through experience and case to case. So human beings, we can understand, we can read the case file and we can actually understand what is the reason or what could be the reason. But AI जो है उसको ये सब, it doesn't have such qualities. What it has alternately is speed. Human being can read a file, maybe 2 files, 10 files in a day. AI will process 10,000 files. So speed is there, scale is there. Scale again is the same, different huge amount of things it can do in a fast time. And then patterns. So AI क्या है कि वो connectivity, वो patterns detect कर सकता है जो human beings नहीं कर सकते. And ultimately hybrid. Hybrid is what? A human being who knows AI, who knows how to apply AI. This is the hybrid model. This is going to give you the best outcome because it is a balance of things. उसमें human का experience भी आ रहा है, उसका जो thought process है वो भी आ रहा है, और computer और data का जो speed है वो भी उसमें incorporate हुआ है. And the decision quality, again final decision जो है human ले रहा है. So that also the trust factor is there. Regulatory bodies को भी पता है कि ये ultimately ठीक है, AI has processed the data, but final decision has been taken by a human being. So that is going to give you the best outcome. So that brings me to the end of the talk. So to conclude on this slide, if you see, in terms of safety monitoring, जो data process करने के लिए, data देखने के लिए जो हमको days लगता था, that time has been reduced to hours by AI, right? Detection of information also is very accurate because you are putting the keywords. And आप लोग अगर कोई डॉक्यूमेंट में कोई वर्ड ढूंढना चाहते हैं, वर्ड फाइल में, सपोज इफ देयर इज अ 100-page वर्ड फाइल, इफ यू वांट टू फाइंड एनी पर्टिकुलर वर्ड, आर यू एबल टू डू इट? 100 पेज के आर्टिकल में मैं आपको प्रिंट करके दे दूंगा और बोलूंगा कि इसमें ये पर्टिकुलर वर्ड ढूंढो, कितना टाइम लगेगा? प्रिंट आउट में without using any technology, if you have to find a particular keyword in a 100-page document, it will take you quite some time. Even with a good reading speed, it will take you couple hours, 1-2 घंटा, 1 घंटा तो लगेगा ही. वही अगर आपको सॉफ्ट कॉपी मिलता है वर्ड डॉक्यूमेंट, how much? Right. So you understand the speed, एंड एक्यूरेसी मिस नहीं होगा. अगर मैं बोलूंगा कि ये वर्ड कितने बार आया डॉक्यूमेंट में, तो वो भी आप मुझे बता पाओगे, बट अगर मैन्युअली करने लगाऊंगा तो इट विल बी पेनफुल. सो दैट इज द हाई प्रेसिजन एंड फास्ट डिटेक्शन. देन डेटा एक्सट्रैक्शन ऑल्सो. डॉक्टर के अलग अलग पेशेंट्स के नोट्स हैं, उसमें से इंफॉर्मेशन एक्सट्रैक्ट करके उसको इलेक्ट्रॉनिक फॉर्म में स्टोर करना, दैट बिकम्स वेरी फास्ट यूजिंग NLP. Then there is this comparison between the traditional and AI driven system which I have already discussed. उसका भी एक summary में यहाँ पे लिखा है. Speed fast है, handling is much faster and bigger data can be handled in shorter period of time. Fine. और manual में क्या है कि expert जो है, the doctor or the analyst who is analyzing, उसके expertise पे focus होता है. Whereas in AI system क्या होता है कि AI based systems में it is focusing on the patterns which are emerging in the input. जो भी articles में pattern है, उसका connections it is able to, AI system is able to recognize those and make a kind of a flow chart कि this has happened, so this is the cause and effect जो बोलते हैं वो pattern बना पा रहा है. AI is able to do that much fast. Then I spoke about the black box. That in AI, in the normal AI, what is happening? Input, output. We don't know what's happening inside in the computer. But with the newer algorithm, XAI जो है, explainable AI, that is helping, that is giving, that output is coming through these different factors. इनका जो contribution है, that is coming through the newer explainable AI platforms. And then what are the barriers? अच्छा AI इतना अच्छा है फार्माकोविजिलेंस में, then we should all shift to AI. We should not do manual, right? Does it happen? No, because there are certain barriers. First I mentioned the data which you are putting in, the quality of the data has to be good. So this who will decide? Again human being only will decide. कंप्यूटर को तो आप 1000 पेपर अपलोड करोगे तो उसको नहीं पता चलेगा कि ये कौन से जर्नल में पब्लिश्ड है, क्या क्वालिटी है, व्हाट इज द स्टडी साइज, व्हाट फॉर इट इज जस्ट अ नंबर. Human being can read it and understand. So data quality is important. Fine. And second, these computational things they require, they are very costly. The softwares are costly and a lot of cost is involved. So ultimately, as I show on the lower right hand, the best way to move forward is in a collaborative fashion. जो human beings का best part है, वो human beings have to contribute, and AI का जो best part है, that we have to use from AI, right? That is the way forward. So thank you all. So any questions? कि मैंने ही पूछ लिया आप लोगों को साइकिल चलाने का और ये है ना, things like that. So if you have any questions, I would be happy to answer. All right. Thank you.
Thank you so much, sir, for insightful and engaging session. It was truly enriching and thought-provoking session. You enlighten us on this futuristic and impactful subject, that how artificial intelligence is transforming drug safety from a reactive approach to a proactive and predictive system. It not only enhanced our knowledge but also inspired us to think ahead in the field of pharmacovigilance. Thank you, sir. As a gesture of our gratitude, I request Mrs. Anshu Dudhe ma'am to kindly present a token of appreciation to Dr. Jignesh Doshi sir. Please, ma'am. Thank you, sir. Now, I would like to invite Ms. Kajal Bondra ma'am to proceed for the valedictory program. Thank you, sir.
Good afternoon, sir. Good afternoon, respected Chief Guest, Honorable Principal Sir, distinguished dignitaries, faculty members, and my dear students. I, Kajal Bondre, Assistant Professor, Adarsh Institute of Pharmacy, Nagpur. After successful completion of 2 days of insightful sessions, meaningful discussions, and knowledge sharing on pharmacovigilance and regulatory affairs in enhancing drug safety, we are now arrived at the concluding segment, that is the valedictory function. A warm welcome to all of you to the valedictory function of National Conference organized by Adarsh Institute of Pharmacy, Nagpur. It is my privilege to introduce our esteemed chief guest for the valedictory function, Dr. Dinesh Chafle sir, Principal, Priyadarshini, JL College of Pharmacy, Nagpur, a position that reflects his dedication, leadership, and commitment to excellence in pharmaceutical section. Now I request Dr. Rupesh Dudhe sir to felicitate our esteemed chief guest, Dr. Dinesh Chafle sir. Thank you, sir. Over the course of this conference, we had a series of insightful sessions that enriched our knowledge and broadened our perspectives. As we conclude this insightful conference, here's a brief recap of overall session. From day one, we began with Dr. Alpana Asnani ma'am, who highlighted the life cycle and safety management of drugs. Dr. Ajay Pise sir introduced the scope of startup in pharmacovigilance, followed by Mr. Shrikant Phule sir, who explained his historical background. Last session of day one, Dr. Omji Porwal sir emphasized the integration of pharmacovigilance with regulatory affairs. From day two, Mr. Shubham Chinchulkar sir focused on strengthening regulatory framework for drug safety. Dr. Darshan Telange sir shared insights on challenges in nano-enabled drug delivery system. And finally, last session, Dr. Jignesh Doshi sir highlighted the role of AI in transforming pharmacovigilance from reactive to predictive systems. Overall, the sessions provided valuable knowledge on drug safety, regulatory advancements, and future innovations. We are truly honored to have such a distinguished personality among us today. I request Dr. Dinesh Chafle sir to kindly address the session.
Respected Principal, Professor Dr. Rupesh Dudhe, Dr. Jignesh Doshi ji, HOD of your Pharmacy Department, Dr. Ms. Shabnam Saher, that is co-convener of this conference, and Professor Mrs. Anshu Dubey, co-convener of this particularly conference. Faculty members, dear participants, and dear students, joining both online and offline, good afternoon to all. Good afternoon. So you have enjoyed, all the students enjoyed 2 days of this conference, or you have taken some notes from the particular lecture or not, or just you listen? सिर्फ सुना है कि आपने कुछ नोट्स भी ली है? ओके, ली है क्या? नहीं. So it is a indeed privilege and honor to be part of the valedictory function of this insightful and timely national conference. Over the past 2 days, this academic platform has successfully brought together intellect, innovation and interdisciplinary collaboration under the one meaningful theme, that is integration of pharmacovigilance and regulatory affairs for the enhanced drug safety. In today's rapidly evolving pharmaceutical landscape, drug safety is not just a regulatory requirement, and it is a moral responsibility. Pharmacovigilance and regulatory affairs are no longer isolated domains, rather they are deeply interconnected pillars that ensure that every medicine reaching the patient is safe, effective and the highest quality. Globally, regulatory authorities such as US FDA or European medicine agencies have established a robust pharmacovigilance frameworks. System like FAERS, that is a FDA Adverse Event Reporting System, and the UDRA vigilance play a pivotal role in a most post-marketing surveillance and signal detection and the risk mitigations. As you know, in India, Central Drugs Standard Control Organization under the Ministry of Health, along with the Pharmacovigilance Program of India, has significantly strengthened the drug safety ecosystem. The establishment of this adverse drug reaction monitoring centers, that is AMCS, across the country has enabled the systematic collections and the analysis of the safety data, contributing to the global pharmacovigilance database such as WHO VIGIREG or managed by the World Health Organization. Integration between the pharmacovigilance and regulatory affairs ensures that safety signals are not only identified but also translated into the regulatory actions such as label changes, risk management plans, RMPS like this, periodic safety update reports, and even product recalls when necessary. So this alignment is critical in maintaining the public trust and ensuring the compliance with integral standard like ICH guidelines, that is ICH E2E for the pharmacovigilance planning. I must commend the organizers, especially the leadership of the Adarsh Institute of Pharmacy, for meticulously designing this conference, conducting it in a hybrid mode as allow a wider participation and knowledge exchange beyond the geographical boundaries, truly reflecting the spirit of the modern education and the collaboration. The conference comprising of the 7 enriching sessions by the eminent personalities has undoubtedly provided the deep insights into the current challenges, innovations, and future direction in the pharmacovigilance and regulatory sciences. Each session has contributed significantly to bridging the gap between the academic knowledge and industry expectation. I would like to add, and the enthusiastic participation in the poster presentation, as I know that there are near about 30 posters will be presented during this 2-day conference, and these platforms are crucial in nurturing the research aptitude among the students and young professionals. They not only showcase the innovative ideas but also encourage the scientific thinking, critical analysis and confidence. Dear participants, as a future pharmacist, researchers and regulatory professionals, you carry a proud responsibility. The ultimate goal of all our efforts is well-being of the patients. In this context, integrating pharmacovigilance with the regulatory frameworks ensure that adverse drug reactions are monitored, reported and managed effectively, leading to the safer healthcare outcomes. You are entering an era of where data-driven decision-making and global regulatory harmonization will decline the success. Understanding the frameworks like good pharmacovigilance practices or good clinical practices is no longer potential, and it is essential. Let the conference not be just an event you attended, but an experience that transfers your perspective. Take forward the knowledge gained here and apply it to to the academic pursuit, research, and personal careers. Before I conclude, I extend my heartfelt congratulations to all the organizers, speakers, and participants for making this commendable grand success and this conference grand success, and your dedication and commitments are truly commendable. I wish Adarsh Institute of Pharmacy continue success in the organizing such meaningful academic endeavors in the future. Thank you all. Jai Hind, Jai Maharashtra.
Thank you so much, sir, for your valuable insights. Now, I would like to invite Ms. Sabnam Sahar ma'am to present a token of gratitude to Dr. Dinesh Safle, sir. Thank you, sir. Now I invite Ms. Minal Raut ma'am to kindly proceed with the prize distribution ceremony.
Very good afternoon to everyone, respected dignitaries, honored guests, and dear participants. Now we move to one of the most exciting segment of today's program, the prize distribution ceremony, a celebration of brilliance, dedication, and the spirit of excellence. Throughout this conference, we have witnessed exceptional talent, innovative ideas, and commendable efforts from the participants. Today, we celebrate those outstanding individuals who have truly raised the conference. I now request our honorable chief guest and respected dignitaries to kindly come forward on the stage to present the awards for poster and oral presentation on hybrid mode. I request the winners to please come forward as I announce your names. Third prize goes to Sharik Sheikh, Karina Nikhare, and Aman Mankar from Adarsh Institute of Pharmacy. They had very well explained their concept through the posters. Now I request Shubham Chinchulkar sir to grace the stage and award them the prize. थांबा. ओके, ठीक आहे ना, जाऊ दे. Second prize goes to Indira Patil from Dadasaheb Balpande College of Pharmacy. The topic of poster presentation was Bridging the Literacy Gap in the Healthcare and the Role of Pharmaceutical Pictogram in Patient Safety. I request sir to grace the stage and award them the prize. एकच नाव डबल आता काय आणि कन्सोलेशन प्राइस हां First prize goes to Lokesh Kade from KC Bajaj College of Pharmacy on the topic Active Learning Based Prediction of Adverse Event Security in COVID-19 Vaccine Recipient.
सर और एक थर्ड प्राइज
एक सेकंड.
नाही
Third prize goes to Dipti Ghate and Palak Nikhade from Tulsiramji Gaikwad Patil College of Pharmacy. Please come on the stage. It was a tie, actually. Congratulations to all the winners for your remarkable achievements. Your hard work, perseverance and passion have truly paid off. To all the participants, your enthusiasm and active involvement have made this conference truly enriching and successful. Remember, every effort is a step towards excellence, and all the participants should collect their respective certificate from the counter. I extend heartfelt gratitude to our dignitaries for their gracious presence and for encouraging and honoring our achievers today. Let us once again celebrate this proud moment with a big round of applause for all the winners and participants. Now, as we move towards the conclusion of this wonderful session, I would like to invite Mrs. Shabnam Sir Ma'am to kindly proceed with the vote of thanks. Ma'am, please come forward and share your words of gratitude.
Thank you so much, Meenal Madam. Respected inaugural guest, Dr. Abhay Tarwar Sir. Respected valedictory guest, Dr. Chafle Sir. Chief Patron, Mr. Gangadhar Nakade Sir. Patron, Devin Nakade Sir. All the distinguished keynote speakers, all the esteemed conference luminaries, Convener, Dr. Rupesh Dudhi Sir, organizing team, faculty members, delegates and my dear students, a very good evening to all. I am Shabnam Seher, today presenting the vote of thanks in the prestigious moment of AIP VIGIREG National Conference on the Integration of Pharmacovigilance and Regulatory Affairs for Enhanced Drug Safety. I express my deepest gratitude to our inaugural guest, Dr. Abhay Tarwar Sir, for his inspiring word and guidance that truly set the conference tone. I offer heartfelt thanks to our valedictory guest, Dr. Dinesh Chafle Sir, for his motivating, constant encouragement in organizing such a meaningful academic session. I would like to express a sincere gratitude to all the keynote speakers, conference luminaries for their enlightening sessions and expert contribution which have gently enhanced our understanding of pharmacovigilance and drug safety. I also extend my sincere thanks to convener Dr. Rupesh Dudhi Sir for its leadership and coordination in making this event a success. A special note of appreciation goes to organizing committee, event coordinator, faculty members and student volunteers whose dedications, teamwork, hard work ensure the smooth execution of this ceremony. I also thanks all the delegates, participants, presenters for their enthusiastic participation and valuable contributions, which made this conference truly interactive and impactful. Lastly, special thanks goes to our associate, that is Prerna Clinical Research, Summer Chemical, and Winspect Business Solution. Now, I conclude my vote of thanks, but I would like to announce the high tea, which is for your refreshment. Thank you so much. Thank you so much.